Hello and welcome to this short segment and presentation of Introductory Microbiology. My name is Ashley Owens. I'm going to be doing a brief introduction of some aspects of genetic modification focused on transgenics. Now, when most people hear the words genetically modified organisms, they think of images of monstrosities spliced together with different parts, beasts that have been horribly mutated beyond recognition, and unfairly transfigured super soldiers made for war. Now, thankfully, the majority of these fears are unfounded and based on conspiracy or overactive imaginations. And although some gene splicing from separate species is a realistic possibility, the way the general public seems to understand it is so far from the actual process, it's actually clouded over into fantasy as well. So, the true science behind genetic modification is not the stuff from some of your worst science fiction nightmares, but in fact, the microbiologists, geneticists, researchers who work in the field tend to produce results that are more like superheroes, if anything. And the wide majority of the goals of genetic manipulation are just like in any other science, aiming to make our lives better. So, researchers around the world are already using techniques of genetic modification to improve agricultural crop yields, developing and improving gene therapy, and even, of course, constantly working to unravel the general mysteries of life of how organisms around us work. So, with all the achievements we've already made using genetic engineering, you may be wondering, well, how much more could we be doing? We've already had major milestones in our research, like unraveling DNA structure and engineering beneficial viruses that can attack and kill deadly disease-causing bacteria. But as most inquisitive minds realize, the scientific community does not work on the old phrase, if it ain't broke, don't fix it. This brings me more along to the lines of the highlight of this presentation, and that's going to be with transgenes. The use of transgenes has been a great game changer for genetic engineering since the late 19th century, and even more so now with our improved technology to help us understand the inner workings of some of the simplest organisms like E. coli and those more complex like fruit flies. But the name transgene comes from the fact that we're transplanting a foreign gene into a different organism's DNA. So generally, we can find a specific protein that produces a desired effect that we'd like hormone production or tissue regeneration, isolate and replicate it, and then use some form of a vector to extract the gene that we need in usually a plasmid from bacteria, and use an enzyme like DNA ligase to splice it back into a different organism's DNA so that the cell will now generate that protein without causing harm. Now, since DNA can be delicate, usually when we're working with organisms, we can't just stick a needle in one organism's DNA and pierce that right into another. We've got several different methods we use. One of the more popular ones, microinjection, but we also have used retroviral methods, and one of the most popular ones, embryonic stem cell mediate transfer. But regarding this, the most interesting transgene that I've come across has been one called green fluorescent protein, or GFP. So this unique protein structure allows it to be integrated into any cell type, and it codes and produces a protein that allows it to fluoresce in blue light. It was discovered from a Pacific jellyfish. So work with GFP gene is almost directly related to a great survival story of a special creature, salamander family called the axolotl. They used to be native only in a small habitat in Lake Chimilco in Mexico, but due to habitat destruction, they have become nearly extinct completely in the wild. But luckily, axolotls have been saved by an extremely valuable medical model for researchers. Among many other unique biological features of the neonatal salamanders, their ability to regenerate nearly any tissue of their body, including bone, organs, and even their spinal cord without any scar tissue at any age has made them an invaluable scientific marvel for regeneration and aging studies. So, in order to better study the regenerative abilities of axolotls, they initially created mosaic individuals from tiger salamanders and our jellyfish protein with the GFP. And once they were able to introduce this into leucistic animals, they were able to hybridize that into a wild-type GFP. So with that model, with these glowing salamanders, researchers were able to watch in real time the processes of the cell's regeneration with GFP tissue that actually could be directly grafted onto different axolotls without tissue rejection by the host animal. And as the research continues, it bodes well as a medical model for even fighting against cancer and aging. So the axolotl is now, thankfully, a living reminder of how genetic engineering is incredibly useful in our lives, and even though this creature was originally nearly doomed, it successfully thrived.
Created using Powtoon.